Alright, so good morning. Despite some difficulties this morning, we're up and running. So the first thing that I did is I showed this 2-minute video, about 2 minute. Thank you, Harvard, for this impressive work. Because I can talk about mutation all day, but literally a 2-minute video of you guys actually seeing it happen, I think really locks it into your brains as to the importance of the impact that mutation of bacteria has on our lives, and how it can mutate very quickly, and especially as it relates to antibiotics. This is bad news for us, right? I looked up the paper. It's some weird antibiotic. It is E. coli. Those are details, right, that sometimes people want to know, but aren't necessarily as important as the overall story that you get from watching that. So that's under our objectives and PowerPoints. It's this video right here, if you care to watch it again or show it to friends and terrify them. This morning, Canvas is being a boob, and I couldn't download my PowerPoints, but I can pull it up, thank goodness, and from my files in Canvas, except, you know, I'm inept at doing that, so even though I have a fancy clicker thing, I don't think it's going to work for this because I'm not in PowerPoint. The good news is that Canvas has a viewer, right? So I'm fast forwarding to where we left off, which is to start talking about mutation. And I wanted to play that video. Damn it. Let's see if it'll work. Woo, look at that. Because we are connected to the internet. So,
how do bacterial cells translate mRNA to proteins? Translation involves 3 types of RNA. 1, mRNA, messenger RNA, provides the code for the sequence of amino acids in proteins. 2, rRNA, ribosomal RNA, is a structural component of the 30S and 50S ribosomal subunits that catalyze protein synthesis. And 3, tRNA, transfer RNA, carries amino acids to the ribosome. Each tRNA carries a specific amino acid and has an anticodon region that complements the codon in the mRNA according to the genetic code. Translation is initiated by an interaction between a specific mRNA sequence near the 5' end of the mRNA, called the Shine-Delgado sequence, and a complementary sequence of rRNA found in the 30S ribosomal subunit. Binding between these 2 complementary regions positions the start codon, AUG, of the mRNA at what will become the P site, or peptide site, of the active ribosome complex. Once AUG is positioned, an initiator tRNA molecule with a complementary anticodon, UAC in this case, enters the P site and binds to the start codon. A special protein called initiation factor 2, or IF2, mediates this interaction. The initiator tRNA molecule carries a molecule of N-formylmethionine, which will serve as the first amino acid in the polypeptide chain. The 50S ribosomal subunit joins with the 30S ribosomal subunit to complete the formation of the active ribosome complex. The active complex has 3 sites that can hold tRNA: the acceptor, A, peptide, P, and exit, E sites. tRNA enters the A site carrying an amino acid. As the ribosome moves along the mRNA, the tRNA next enters the P site, where its amino acid is added to the growing peptide chain, and then moves to the E site, where it is released from the complex. Now, elongation of the polypeptide chain can proceed. At the start of elongation, the A site, or acceptor site, is empty. An amino acid carrying tRNA molecule with an anticodon that is complementary to the mRNA codon enters the A site and binds to the mRNA. As the ribosome moves along the mRNA 1 codon at a time in the 5' to 3' direction, a peptide bond forms between the N-formylmethionine and the amino acid carried by the tRNA entering in the A site. The new dipeptide.
So now I'm going to fast forward her. You get the idea, right? So it's just going to keep moving along, right? Transfer RNAs transfer in the amino acids. They join to the joining train until it gets to the end where it reaches, very important, stop codon, and it stops. This is the next part. Wait a minute.
While it is still
This is in a bacteria. So this is something unique that happens in bacteria.
mRNA is available for translation into polypeptides while it is still being transcribed. This is possible because bacterial DNA and ribosomes are not separated by a nuclear membrane, as they are in eukaryote cells. When a stop codon enters the A site of the active ribosome complex, it is recognized by two special
picture. Let's see if I can get it at the right spot. Right about there.
DNA and ribosomes.
Pause, Erica, pause. That's good. Okay, so this is the DNA, right? So it's double-stranded, right? So notice the two different blues. And this is RNA polymerase that is attaching to the DNA and is making the messenger RNA, which is the purple, right? The transcript. So even as it was making it, right, as soon as there was enough available and that start codon somewhere on here in the Shine-Delgado sequence was available, the ribosome formed on it, and look, the protein that started being made, right? So this process is not separated. In our cells, in eukaryotes, it's separated. Messenger RNA is made in the nucleus where the DNA is. It leaves the nucleus and goes to ribosomes either in the cytoplasm or embedded in what organelle is rough because it has ribosomes. Right, the endoplasmic reticulum, right, has that rough appearance because it has ribosomes there. So, and as we saw, and I spoke about last time too, sometimes it'll produce such a long transcript, multiple ribosomes are going to attach, and multiple different proteins can even be made, right, from one big long transcript of messenger RNA. And this is typically done when they're making the enzymes safe for a pathway, like glycolysis or the Krebs cycle, right? They're going to make all those proteins at the same time, right? And they can start a process before the other one even ends, which is kind of cool. But that's unique to bacteria, to prokaryotes. We don't do that. We do a whole lot more in eukaryotes. Eukaryotes are more complicated. We don't have a single chromosome either, right? We have multiple linear chromosomes usually. All right, so let's see if we can get to the PowerPoint. Where do I have to go? I have to go to my files. There we go. All right, so make it back big screen again. All right, and I have to click these. Isn't this fun? No. Okay, so we talked about how we know this code already, right? So that as they were coming in, those transfer RNAs, they know the messenger RNA code. They know what amino acid to put there. But what was one of the things that was important when we looked at this picture? So there's a start codon and there's 3 stop codons, right? But look right here. This is leucine, right, which is amino acid. This is the codons that code for leucine. There are 4 of them, right? The first 2 letters are the same. The third one really kind of doesn't matter. Do you see that? There's what we call redundancy in this code. And that affects the end product, because remember, DNA changes, that's called a mutation. If the DNA changes, it's a mutation. If the DNA changes, the messenger RNA changes, yes? When the ribosome is reading that messenger RNA, is it always, because there's a change in the DNA and the messenger RNA, is it going to put a different amino acid there? No. Depends on where the change happened, right? Often when it's in the 3rd position, it may end up being the same amino acid. We still call it a mutation, right? Because the DNA still changed. The only difference is the product didn't change. You still made the same protein. You still with me? Does anyone gander a guess what type of mutation that's called? Because it happened if we talk about one nucleotide changing, right, one of these bases, that's a point mutation, right, where one thing got changed. But because the amino acid didn't change, it's called what we call a silent mutation. So as I said, there's redundancy, right? We have 20 different amino acids, but we get 61 different codons for those 20 amino acids, right? So we have some redundancy in that code. We have 3 stops and 1 start. So as you saw in that video too, right, this Shine Delgado, I always gobble that last name, right, is where the first part attaches. That's why there's RNA in a ribosome, right? So it can recognize the messenger RNA. So a similar process happens in our cells. And the same thing for us. It's AUG. It's the same code, but there are a little bit differences between prokaryotes versus eukaryotes, but not very many. And so this is what I was alluding to earlier, right? We saw this happening, right, where the messenger RNA was being produced and the ribosome was binding and the protein was already being made. But they could actually have, what they didn't show you there, multiple proteins all being made at the same time. And that's unique to prokaryotes. And then the folding of the protein, right, as I mentioned, we're still trying to figure this out, right? How is it that once we make that big long of amino acids, how is it that these interactions happen and fold up as they do? But we do know that this structure, this globular tertiary structure, is very important to the functionality of a lot of the proteins, especially ones that are enzymes, because that's going to determine its active site where it interacts with the substrate. So think about this, especially like look at this picture right here. See this disulfide bridge that's happening right here? That's because these 2 amino acids have sulfur in them. And there are actually only 2 amino acids that we have that. One of them is lysine. No. Oh my god. We're going to talk about them later. I'm not going to name them. I don't want to be wrong this morning. My brain is not coming out of the brain. But think about this. If we switch this amino acid for an amino acid that does not contain sulfur, is this disulfide bridge going to happen? No. And so that's going to change the structure of that protein, right? Could it make that protein no longer functional? Absolutely. Right? Where, say, this amino acid right here next to it changes. Well, look, it's not interacting with any other amino acid in this secondary structure we're looking at. And maybe it doesn't interact with the substrate, so maybe that change is no big deal. Has no effect on the organism, no effect on the folding of the protein or the functionality of that protein. What if, look at how long this, so say this is the start and this is the end. I don't know which is what, but let's just for sake of discussion, right? This is the start of it, the big long chain of amino acids, and this is the end. What if we stop right here? Like we get a mutation that happens and a stop codon gets put here. This whole section is not going to be made, right? Again, is that section of that protein important? Possibly, depends on the protein, right? So when these changes happen, sometimes they can be really bad, basically make the protein no longer function. Well, if you're an organism and you need this protein to function for life, you're going to die, right? That's going to be a lethal mutation. Those don't get passed on, right? The organism died. But sometimes they're minor mutations and don't affect the functionality of the protein. So they get passed on. Or sometimes what happens is what I call winning the genetic lottery. The enzyme is better for some reason. Either it functions faster or better, or in the case of bacteria, I don't remember the target for that video that I watched of that particular antibiotic. But some antibiotics interfere with protein synthesis by literally binding to the ribosome. So remember, the ribosome is made out of RNA and protein. So what if that protein component changes? And that spot is, say, right here, is where the antibiotic binds and therefore interferes with protein synthesis because it binds to the ribosome. Well, what if a bacteria evolves and changes this amino acid? Is that antibiotic going to bind to it anymore? No. Right? It's got to be able to literally physically interact with each other. Antibiotics literally bind to things within the cells and interfere with their functionality. That's how antibiotics work at their core. So if a change happens and that antibiotic can no longer bind to that protein, it can no longer interfere with that bacteria, and that bacteria is said to be resistant to that antibiotic. So that's what you saw happening in that first video, right? Is that bacteria are constantly changing, and they change at a faster rate than we do. Do you guys remember how long it took for them to get to the middle of that petri dish? Who was paying attention? 11 days. Right? And that was, we were basically watching natural selection, right? Because as they evolved, if they evolved a change that allowed them to be resistant to that antibiotic, they were able to continue to grow. All the rest were killed off by the antibiotic. And that's what happens in our world today, right? Bacteria are constantly changing. And so we're going to get to the point we've already reached it. There's a bacteria out there resistant to all known current antibiotics. Right? The bacteria don't change so that they're resistant. They change because of this process is not a perfect process, right? They make mistakes when they're copying the DNA. Under that selective pressure of the environment, though, that determines whether that change is good or bad, right? So they're not like, oh, I'm exposed to this antibiotic, so I'm going to change this part of my ribosome so that I'll be able to survive. No. That's why I kind of equate it to the lottery, because they're basically, you know, they're changing, and it's kind of just luck of the draw, whether it's a good or a bad thing, that change. Does that make sense? Like, you don't know when you pick those numbers if it's going to be good or bad, right? And once you've picked, you can't go back and be like, you buy more tickets, but you get the premise here, right? The bacteria are changing, and it's the environment that determines on whether that's a good or a bad thing. Even the environment within the cell. That's an important point to understand. Okay. So let's look at some specific mutations, right? So we're going to look at what's called base substitutions. So when we talk about DNA, remember what makes each nucleotide different is that nitrogenous base. The A's, the T's, the G's, and the C's, the adenine, the guanine, the cytosine. Remember those? Right? So when we say base, that's what we're talking about. We're talking about changing out one of those nitrogenous bases. The adenine, the guanine, right? Cytosine. Just changing one, right, can have a substantial effect on the cell. It could result in a silent mutation, which that one kind of implies what I was trying to get out of you guys earlier, is that just because the DNA changes, does it mean that the amino acid's going to change or the protein? No. But a mutation still happens because the definition of mutation is what has to change? The DNA, which is the basis. Okay? So the other things that can happen is a missense mutation or a nonsense mutation. Anybody yet remember which one of those I called stop codon? missense and nonsense. Nonsense is another name for a stop codon. Codons that code for something, for an amino acid, are called sense codons. So missense, in its names, tells you, guess what? There was a mistake that was made. So we're going to look at examples of this. We're going to look at examples of frame shift. This is a different type of mutation that can happen. We'll look at the other ones later, because they're unfortunately another chapter, which are transposons and knockout mutation. We're going to look at induced and chemical mutations today. We're going to talk about a specific example of a chemical mutation, athenium bromide. We're going to look at the effects of radiation, UV light, and X-rays today. So, I hope, right? All right. So, in general, DNA replication occurs without much error. But, of course, mistakes happen. Those mistakes we call mutations. And as I've already been talking about, right, they can be bad, but they're not always bad. And when they're not bad, they're why we see evolution, why we see change. With the pressure of the environment determining what is good and what is bad. And of course they can alter it and therefore affects its function. But I find it's best when we have like real examples, which we'll talk about later. So definitionally wise, and we'll look at some pictures to go along with this. A silent mutation usually happens in that third codon position, because as we looked at that code that we know, it's really that third position doesn't matter as much as the first two. So if the third base in that codon gets changed, sometimes that's not going to result in a different amino acid. So we get the same protein, like unless you do genetic testing, right, sequencing, you don't even know that that mutation happened, right? Hence why it's called a silent mutation. There's no change in the protein, but there was a change in the DNA. A missense mutation, on the other hand, is a change in a codon, so probably either in the first or second position, and a different amino acid gets put there. Again, it depends on how important that amino acid is to that protein as to whether that's going to be good or bad or lethal. Nonsense mutation is where it's supposed to be an amino acid, but instead a stop codon gets put there. And typically this means stopping making the protein before you're supposed to stop, right? So you're going to get a shortened, abbreviated protein. What's the chances that that protein's going to be functional? Not very likely. So if you were to rank these from like worst possible situation to best possible situation, worst, nonsense mutation, then next would come missense mutation, and the best is a silent one where it doesn't even change the protein, but the DNA changed. All right, so let's look at some examples of that. All right, so I'm going to zoom in on each one of these. The wild type, right? Oh, this is not the PowerPoint I want. I thought I zoomed in on them. Okay, I guess not. I'm not going to risk going back to my files to open a different one. This is an older version. That's okay. Most of you guys have computer screens or you printed it out, right? And the computer screens are closer to you than they once were. So you can see. Wild type means normal, right? The most commonly found sequence for a particular protein within an organism, right? That's what wild type means. That means that's the normal. right? No mutation. Okay, so what they show you here is that right here, this one base, right, that is a T is replacing a C. So if you look up in here, right, it's supposed to be a C, G, but it got changed to a T. So now it's TA, which changes this to a U. But the amino acid that's supposed to be there is arginine. It's still coding for arginine because it's in that third position where the change happened. So we call that, of course, silent mutation. Protein didn't change, DNA did though. But it's that whole step of translation, right, that kind of determines what change is going to happen. So here on this next one, right, an A gets replaced with a C, and it's at the first position. So instead of arginine being put there, it's serine. So we call this a missense mutation. Again, it's really going to depend on the organism if that last amino acid being changed is going to make a difference or not. Right? What kind of effect that's going to have on the cell. But as far as the mutation goes, we call this a missense mutation because a different amino acid got put there. The worst case scenario, of course, this change that they show happening resulted in it made one amino acid and then it stopped. There's no way that's a functional protein, right? One amino acid. It's not going to do anything all by itself, right? This is, of course, what we call a nonsense mutation because a stop codon was replacing what should be an amino acid. And, of course, a stop tells the ribosome to do just that. Stop. They're very much like computers or robots, right? They just follow the code. Right? They're not going to analyze it. They're not going to go, oh, that's not supposed to be there. They're going to do exactly what it says. So for base substitutions, if you just switch out one base, these are the 3 different possible types of mutations that could happen. Silent, missense, or nonsense. From just changing one amino acid. So that's changing an amino acid. What if we delete or add amino acids? Remember, it's a code that's read by the ribosome in groups of 3 nucleotides that we call codons. Right? 3 bases. So in this case, right, they added, I think, right? I wish they would tell me. I think they added this one. Yes, they added it. So notice we've got our start, AUG, for methionine, AUG, then AAA, AAA, and instead of GGG, they put a C in there. So now it's CGG, and so now it's a different amino acid. And what happened to the rest of the code? It's supposed to be right after GG, CC, but now it's GCC, and then CUU. It completely shifted that code, right? So from that point on, look at, we also have different amino acids. So when you add or you delete 1 or 2, even 3, depending on where it happens, you're going to change that code from that point forward. So although in my class, I don't use these horrible things anymore called scantrons, most of you have experienced taking a test using a scantron, right? And this is the perfect example of how forgetting or adding an extra letter on that darn piece of paper, right, could have quite debilitating effects on your results. Because, right, if there's a bunch of A's in the row, right, and you put an extra A, that shifts all your answers on your Scantron sheet, right? So even though maybe you're getting them right, you're putting them in the wrong position on that Scantron sheet, right? And unless you realize it, right, and when you do, depending on how far along you are, you're erasing a whole bunch of stuff to get it all back right. Okay, order matters. Order matters, right, for our messenger RNA. If you add something or delete something, you're going to shift that order from that point forward. The Scantron analogy helps though, huh, right? You've all probably been in that position at one time or another and realized how bad that can potentially be. So as you can imagine, as we talk about mutations, this is probably the most damaging, right? The one that usually results in non-functional proteins, right? Because it's just too much change, right? And of course, it really does depend on where that change happens. But from that point of the addition or the removal, the whole thing changes. So let's see. I like the Scantron better, but in the past I used to try and use words. So don't make fun of my grammar or the context. But these are all three-letter words, right? The dog ate the cat, right? If I make you laugh, sometimes you remember things, though. Okay. So in this next sentence, what happened? I added an A, but I did just like a ribosome would do. 3 letters is a codon. In this case, 3 letters is a word. I just messed up that entire sentence, didn't I? By adding that 1 letter. The next one, what did I do? I deleted the first letter, but then did just like a ribosome would do. 1 letter, right, is 1 base. 3 groups is a codon, equates to amino acid. All right, so the big dog ate the cat, right? In this case, I added a whole codon, right? If you were to talk about it in DNA language, did I mess up the sentence? No, right? This is the one exception where adding or deleting will not result in a frame shift. But this is if it happens in between codons, right? If you take a whole codon and stick it in between codons, or you remove a whole codon. So, if you remove or add a whole codon, what are you removing or adding as it equates to a protein? What are proteins made out of? Amino acids. So this is the one exception to frame shift, where an adding or a deletion won't create a frame shift. As if you take a whole codon, stick it in between codons, right? You can't put it within a codon, that would still mess it up, right? Create that shift. It's in between. This is rare, but of course it can happen. This is definitely my old PowerPoint. Alright, so other funky things can happen that definitely can happen in our cells because we have linear chromosomes, right? And of course they do this on purpose to try and exchange information and create variability. Is that during one of the phases of mitosis or meiosis, right, when we're creating our sex cells, the 2 chromosomes that match each other will connect with each other and switch information. But the information on either one of them is very similar, right? But there's different variants. We call these alleles, right? Remember that term? Right? So probably the best example to give you a real world example that you can wrap your brain around is blood typing, right? So for us, there's the ABO blood group, right? You either have the A antigen, the B antigen, or you don't have either one of them, which is O, which by the way was supposed to be 0. Not O. It's meant to be 0. Meant to say you don't have A or B. So but think about this. You get one chromosome from mom and one from dad, right? And maybe one's got A and one's got B, right? And during this process, maybe we switch them up. And it's just random as to which one you give to your kids too, right? Whether you give them A or B. But what's happening here in these pictures? Look at this first one. When the information is being crossed over, so each letter represents, say, an allele or a segment of information. It got flipped. You see that? That's not good. Because as we know, enzymes only read information in one particular direction. So notice that it says in big bold letters, very damaging to the cell when it goofs like this. Typically lethal. Or in this next picture, what's happening? The wrong chromosomes are exchanging information, right? Certain chromosomes have certain information. If the wrong ones exchange information, then there's a possibility of loss of information when those split from each other. Because remember, they're going to divide up. One copy goes to one and goes to the other. So this is an inversion, this is a translocation, right? It's being located to some other place. All right, so how is DNA made? Right, so you have DNA and you're going to copy it. The name of this enzyme is really easy, y'all. It tells you what it does. It's called DNA polymerase. Right? And as it's copying that information, and it literally like hooks onto the DNA and slides down it. So it structurally is holding on to it. And so as it's making these copies, remember when I talked about how beautiful that spiral staircase is and it's like 3 rings across all the time? You got purines with pyrimidines. But what if something comes in and it's not supposed to match? Because of the shape of that enzyme around it, sometimes it won't even go in, or it'll go in, and as the enzyme goes to move, it gets stuck because it's the wrong amino acid that it just put in there. And so what this enzyme has the ability to do is, as it's making it, it kind of proofreads it. So especially things that would make it kind of bulky and not that beautiful spiral staircase, those types of mistakes, it can fix very easily. But DNA polymerase itself, what makes DNA, can also proofread it. So just like you, right? You can proofread your own work, right? Are you perfect at it? No, right? You sometimes miss mistakes. So sometimes it's going to miss mistakes. But it does have ability to fix its own mistakes. So DNA polymerase, right, has an error rate of about 1 in a billion. Boy, I wish I made a mistake 1 in a billion times I did something. So this is very, very low, right? And in part is because of that proofreading ability of DNA polymerase. Right? It can really recognize its own errors and replace them. But again, they do make mistakes. Not very often, but often enough that change happens. Mutations can be induced. We can either do this in the lab, or this can happen in nature due to chemicals, right? And this is our biggest fear, right? Is what are chemicals doing to our bodies? More importantly, too, what are chemicals potentially doing to our DNA? So adenine right here can be chemically modified into this different nitrogenous base. Adenine's supposed to bind with which one? What's the base pairing? Thymine. But in this case, this modified one is binding with cysteine, right? So we're going to end up with a base substitution at that point where that change was made, right? By no means do I, of course, want you memorizing any of this. The important thing is here is that these bases can change, and their base pairing changes, and therefore the code is going to what? Change. It's going to get mutated. So a chemical mutagen we actually use in the lab, and why scientists started using this one wasn't because it mutates DNA. It's because it binds to DNA and fluoresces under UV light. So when you hear scientists talking about gel electrophoresis, right, and things like that, what they've literally done is they've taken DNA because it's highly negatively charged, and they stuck it into this gel matrix, right? So it's like a sponge with holes in it, and there are wells at the top, and they put the DNA in the top, and then they apply electrical current to it. And so depending on the size of the DNA, it will head down, right, this gel, down this sponge. Because it's got holes in it, right, in between the molecules. And of course, the really big pieces of DNA stay towards the top, and the smaller ones can move through more of the holes, and they'll migrate further down. What you see here is what's called a ladder. It's a standard. They know what the length of each one of those lines is. So this is one of the ways that, because of course DNA you can't see with the naked eye. This is, you know, thousands of pieces of DNA actually. But there's enough there that with this athenium bromide that literally binds to the DNA, it fluoresces under UV light. So that's why scientists use this, to help them visualize DNA, very large pieces of DNA, in a matrix, right? And so when they were cutting up DNA, they could very easily see how many pieces they ended up with, right? And about what size they were, right? This type of, you know, tagging is even done in really high-tech gels where they're sequencing, like literally looking at each individual. They put little markers that are different from athenium bromide. The problem with athenium bromide, and you won't see a bottle of it here at Delgado because we're not cleared to work with this high level of agent. I worked with it when I was in graduate school. The bottle had on it a skull and crossbones. Right there makes you like not even want to pick up the bottle, right? And it says on it possible mutagen. No, it says mutagen, possible carcinogen, right? Because if you change your DNA, you could potentially change it in such a way that the cells become cancerous. So always wear gloves. We always dispose of this in a very safe manner. I had a colleague of mine, he never wore gloves. I'm like, what are you doing? I'm like, you're crazy. You know, he's like, oh, he's outside skin cells or dead cells anyways, you know, it's not going to get into my body. I was like, yeah, I want to have kids. I'm gonna practice safe practices when dealing with this, because if it incorporates into your DNA, right, it's called an intercalating agent because it does just that. It sticks to the DNA. And so remember that beautiful spiral staircase I talked about how like the length, right? So the length across here is always, say, 3 feet. In this case, what is changing? These are the steps of the staircase, right? So all of a sudden, right, every step is 1 foot up, right, say? And then you get to a spot where there's like this 2 foot gap in between steps. Of course, you're on the phone to the carpenter and you're like, what happened, dude? You run out of materials or something? Go put my step back in, right? I'm not going to hop up, right, 2 feet to the next step. That's ridiculous. And DNA polymerase, when it's coming along, either it's going to stop, right, because this is an obstruction, or it goes, there's supposed to be a step here, so I'm going to put a step here, right? So they're going to add a base, right? He fixed it. It's beautiful, right? But, right, is there supposed to be an extra nucleotide right there? No, what type of mutation is this going to cause? A frame shift. Which, of course, is not good, because from that point on, DNA has changed, right? So you can see why I wore my gloves, right? I didn't want any frame shifting happening to my DNA, even potentially on my hands, or anywhere else for that matter. So this is one we're exposed to every day that you walk outside. It's UV light, right? It can cause your thymines next to each other in a DNA strand who are supposed to be hydrogen bonding with their friends adenine across the way, right? You see that? Instead, they're covalently bonding, which means they're sharing electrons. That's a strong bond, right? Hydrogen bonding is what happens between the two strands, and that's a little bit easier to break, right? And so that we can pull apart the two pieces of DNA and copy them, right? But what if these guys are covalently bonding with each other? Okay, all right, so they're bestest friends right now. Who cares? Look at the DNA structure. It's been distorted, right? How does DNA polymerase or even RNA polymerase read it? Literally hooks on to the DNA. Imagine a train going down the tracks. Gets to a buckle like this. We're talking about derailment, aren't we? Not good. So what happens to DNA or RNA polymerase when it gets to this? Sometimes it will derail, right? Just stop making that protein. Or stop copying that DNA. Or what's another thing it could do? Misread that, right? Maybe it sees it as just one T. What did we just do? Deleted a T, right? Yeah. But this is a different type of change, right? We've literally distorted, right, the structure of the DNA. So again, you're lucky if it even reads it. And if it does read it, the possible mistake is it reads it as just one T instead of two T's. And that's going to create what type of mutation? A frame shift, right? Because we're deleting, right, information. And so then we're going to shift it from then on. So now we're all terrified to go outside because guess what? This is what's happening to your DNA when you go outside. Does it stay like that? No. Another reason why some mutations do not stay or even happen is because we have repair mechanisms that recognize it. So DNA polymerase, yes, can proofread, but we've got other systems behind that checking the DNA as well, right? So for this one, right, we have enzymes that can break this bond and put this and get these guys to be best friends again, adenine and thymine and hydrogen bond, right? And then we have the beautiful spiral staircase again. So we're going to look at those, yes, today. Good. We're going to look at those. So what do you need to know? You need to know the general pathway and what it fixes. So we're going to look at what's called photoreactive pathway, which another name for it is direct. And there's a reason you'll see why it's given this name. It works directly, and it uses what in the process? Photoreactive. Light. And there's another clue in there that helps you out, y'all. This fixes damage that light causes. UV light. So that one's easy. Mismatch repair. What type of mutations do you think that fixes? Missense, nonsense, silent, when we have a mismatch, right, of the DNA, right? When they don't match up, right? When you have a base substitution, right, which can result in silent or missense or nonsense mutations, but we're not even going to get any of those, right, because we're going to recognize that there was a mismatch that happened. So how do these mechanisms work? That's what we're going to look at next. So your book lists this. We're not going to go into that one. We're just going to stick with mismatch repair and photoligase, or direct repair by the photoreactive pathway. And then we've already mentioned that DNA polymerase has the ability to proofread. So it reads 5' to 3', but it can back up. And that's what they mean by 3' to 5'. Exonuclease. Exo means to remove. Nuclease refers to the nucleotide. So it literally will back up and remove the one it put in and move forward and put the right one in. So that's pretty easy. And then they go in a whole lot of detail, right, for mismatch repair. We're going to keep it basic. All right, what's important here? This strand of DNA, and this is a strand of DNA. This is the mismatch. C with T. Okay, we have a problem though, right? Which one's the right one? We have to be able to distinguish which is the correct nucleotide, right? The C or the T. So the way that our cells do that is that as our DNA matures, something gets added to it. You see this? Those are called methyl groups. Our DNA is methylated. So that tells the system this is the template strand, right? This is the original strand. This is the correct code. This is the new strand. This is the wrong base. It needs to be removed. So we have special enzymes, right? They're going to read, they're going to detect this mistake. They're going to attach, and they're literally going to cut out that section of DNA where the mistake is. They don't just take out that one nucleotide, they cut out a whole section. It's the way they work. Right? So it's kind of complicated, right? The process of actually cutting it out. But the important thing is they cut it out, right? So, oh no, what needs to happen? DNA polymerase needs to come along and put that piece back. And then the pieces have to be joined back together, right? So this piece that they put, right, these pieces have to be joined. So that's what DNA ligase does. The name tells you, right, what it does. It puts pieces of DNA back together. It repairs that phosphodiester bond, the sugar phosphate bond that makes up the backbone. So lots of details here in these pictures, right? What's important? With mismatch repair, enzymes literally recognize the mismatch because they know which strand is the old and which one's the new, right? They're going to cut out the mismatch and the new. Enzymes are going to replace that, and then we're going to glue it back together. DNA ligase. Problem fixed. Yay. No mutation. Alright, so we've already said this buckle, this thymine dimer, can be caused by UV light. So we get this buckle. So enzyme called photoligase, right? Because what does it do? And why is it sometimes called direct repair? Literally binds to DNA. Literally uses light energy to break that covalent bond between the two thymines. So they'll go back to hydrogen bonding with their dear friends adenine across the way. So light causes it and light fixes it. So and then it leaves. It's fixed. Problem fixed. Okay, which gives me a little bit of time to pray. It's the end of our stuff for chapter 7. See if I can get back to Canvas. Who knows, right? It's behaving. So notice right here, this PowerPoint is our poll quiz for next time. So you can look at it ahead of time. You can print it out ahead of time. And I gave you all a homework assignment because I wanted you to learn your login so you could log into these computers, because your login is your email, right? And your email password for your Delgado email, right? So you still haven't done that assignment yet. You can do it. It's worth 5 points. I would do it soon because this week is considered midterm, right? The good news for you guys is I don't do a midterm exam. We've already had an exam. We're not going to have another one until we finish this material, which is set for October 23rd. I hope to stick to that deadline. We'll see if that happens. Really depends on, you know, environmental factors we have no control over, right? So what I'm going to be doing before the end of the week even is I'm going to be adding a column that says midterm grade. And so if you haven't done assignments, zeros are going to be getting put in for any Module 1 assignment that you didn't complete. So I highly suggest if you didn't complete it, do it now before I go in and put zeros, right? I grade it instead. Those zeros can be erased if you do those assignments. But note that as soon as I put your midterm grade in, I'm not going to change it, okay? So, and midterm grades at Delgado, right, are our internal system thing. They don't go on your transcript. So if I make a mistake even, I can't fix it, okay? I double and triple check final grades because those are important. Those will go on transcripts, right? If I make a mistake, I have a way of fixing it. Midterm grades aren't that important. And they're mostly from when, before the computer age, where you guys were always knowing where you stand grade-wise. But I'll explain if you're not familiar with that. But you can't right now because I don't have everything deployed in Canvas. But you can kind of play around. But know that your tests are the bulk of your grade in this class, right? But assignments are going to help you. If you don't do your assignments, that's going to bring you down about a letter grade. If you do your assignments, it brings you up about a letter grade from your tests, because they're 25% of your overall grade. And if you do them in a timely manner, they help you prepare for the exams. Thus, you get a higher grade on the exam, right? See how these things work together? Okay? So, know that I'm going to be doing that soon. I'll post an announcement, right? The important thing is you're here. But if you're a student that haven't been coming and you haven't been coming to class and you get an F, you actually get what's called an FN. If I can't calculate a grade from you because you haven't done any assignments, if you get FNs in all of your classes, you get booted from school because you're clearly not coming to school or participating in school. If you get an FN in a class, that teacher can, you might as well just drop that class, because if the teacher doesn't allow you to make up those assignments that you missed, you're going to get an FN at the end of the semester, right? I'll tell you, for me, it's situational, but most of you guys have been coming to class. It's kind of like, you know, talking to people about coming to church to the people who come to church. But it's important information you might want to disseminate to your friends or know for the future, right? You need to come to school, right? If you sign up for school, you need to come for school. There are consequences if you do not come to class. In this class, there's an attendance grade, right? And that was one question that somebody asked me too. I don't know if that grade column is showing up, but at the end of the semester, you're allowed for me 2 unexcused absences. You don't even have to tell me why you weren't here. And I will adjust your grade, your attendance grade. I'll give you points in a separate column to make up for those 2 days you missed. Okay? Make sense? I don't do it till the end of the semester. Right? That way, too, you know, you see, right, your attendance grade. You know what days you missed, right? And I'll have to put today's in, and where is today's attendance sheet? Make sure that you sign it. Can you put it on the front table? Did anybody not sign it? Okay, we're good. I just need to make sure I get that so I can put it in for today. And don't be late, and try to come early. I'll be early. I had a doctor's appointment this morning, which is why I was running late, and was very upset that this system wasn't working when I walked in. But please come in and sign on to use the computers, because as you know, there's like no Wi-Fi in this room. So doing texting and doing our polling is going to be really difficult, and we have the computers, let's use them. Right? But I'm not running around and signing everybody in. Right? You got to use your login. You got to log out, right, of these computers, please, when you get done using them. Okay? If you don't know how to do that, I'll help you. Okay? Any questions from me? All right. Oh, look, I see I still have 3 minutes. I'm good. All right, y'all. Have a good one.